When you think about the biggest challenges in modern medicine, fighting brain cancer has gotta be right at the top of the list. But what if? What if we could build a realistic, living model of a brain tumor outside the human body? A place to test new treatments safely and effectively. Well, that's exactly what researchers at TU Delft have done, and we're about to dive into their incredible breakthrough.
But before we get to the solution, we really need to understand the problem. It all boils down to one central question. What is it about the brain that makes it such a uniquely challenging battlefield for doctors?
And the answer, well, it has to do with a remarkable and frankly incredibly frustrating piece of our own biology. It's the brain's very own personal fortress.
Let me introduce you to the blood-brain barrier, or the BBB for short. Now, I want you to think of it as the most exclusive, VIP-only nightclub bouncer in your entire body. It's this tightly woven network of specialized cells that lines the blood vessels in your brain, and its job is to meticulously control what gets in and what stays out.
And this is where you see the real double-edged sword. On one hand, the BBB is a brilliant protector. It shields our most vital organ from toxins and keeps its environment perfectly stable. But on the flip side, for medicine, it's a massive obstacle. It blocks potentially life-saving drugs from ever reaching a tumor, and it makes it almost impossible to study how those tumor cells are behaving in their natural habitat.
So, if you can't easily get past that barrier in a living person, how do you study it in the lab? Well, for decades, the methods we've been using, let's just say they've given us a pretty fuzzy and incomplete picture of what's really going on. Our traditional tools just haven't been up to the job. I mean, growing cells in a flat petri dish, that's kind of like trying to understand a skyscraper by only looking at a two-dimensional blueprint. You miss all the complexity, all the crucial 3D interactions. And while animal models are definitely important, a mouse brain just doesn't have the same biological setup as a human one.
So here's the bottom line. To fight a complex enemy like this, you need a complex and, more importantly, an accurate map of the battlefield. Researchers didn't just need a better model, they desperately needed one that was much, much closer to the real thing.
And that is where this brilliant innovation from a team at TU Delft and Erasmus MC comes into play. They basically decided that if you can't easily study the real thing, well, you're just going to have to build the next best thing. Their tool of choice sounds like something straight out of science fiction. It's called two-photon polymerization. So imagine a 3D printer, but instead of squirting out melted plastic, it uses these incredibly focused laser beams to solidify a liquid gel into microscopic, super intricate shapes. It lets them build these unbelievably detailed structures, layer by tiny layer.
And the process itself is just so elegant. First, they print these tiny scaffolds. Think of them like microscopic apartment buildings ready for cells to move into. Then, they add human blood vessel cells, which form the basic foundation of the barrier. Next up are pericytes, another kind of cell that helps keep everything stable. And finally, they introduce the enemy, aggressive glioma tumor cells, and then they can just watch the invasion unfold in real time.
Now, this is where things get truly groundbreaking. You have to understand, getting different types of human cells to just live together happily outside the body is insanely difficult. But for the very first time in a model like this, they managed to grow 3 distinct types of human cells together, all interacting naturally on one of these tiny 3D printed scaffolds.
And the results? They really spoke for themselves. As the lead researcher, Angelo Accardo, put it, the cells grew in a way that was much closer to how they'd act inside a living body, the in vivo situation, than anything seen in a flat culture. The model worked. When they added the tumor cells, the barrier broke down, exactly like it does in patients.
But this team wasn't satisfied with just having a static 3D model. I mean, our bodies aren't static, right? They're filled with constant movement and flow. So, they decided to take their incredible creation and push it one giant leap further.
To do this, they took their mini-brain environment and integrated it onto something called a microfluidic chip. And this is the step where the model truly comes to life.
So what in the world is microfluidics? It's actually pretty cool. It's essentially plumbing, but on a microscopic scale. By putting their 3D cell model inside a chip with these tiny little channels, they could actually pump fluid through it, perfectly mimicking the constant flow and pressure of blood running through our veins. And adding this dynamic flow is a huge deal. It puts real mechanical stress on the cells, just like what happens inside the body. It makes the entire system an even more faithful replica of what's happening inside a real brain. It's really the difference between looking at a static photograph and watching a full-motion video.
So,
What does all of this actually mean? What's the big takeaway? Well, this isn't just some clever lab experiment. What we're looking at here is a powerful new platform that could fundamentally change how we study not just brain cancer, but a whole host of other diseases.
The platform they've built is a true game changer for a few key reasons. First, it's robust, which means it's stable and reliable enough to run long experiments. Second, it's reproducible, so labs all over the world can build it and get the same results. And maybe most exciting, it's incredibly adaptable. You could pretty easily swap out the brain cells for, say, liver cells or kidney cells and use this same approach to study a whole range of different conditions.
And they're not stopping here. The team is already planning the next upgrades to make the model even more realistic. Next on their list is to add microglia, which are the brain's own specialized immune cells, and astrocytes, another type of cell that's crucial for keeping that blood-brain barrier strong.
And all of this incredible science, it all leads to one final, really powerful question. Just imagine a future where doctors could take a tiny sample of a patient's own tumor cells, grow them on one of these chips, and then test a dozen different drugs to see which one works best before ever giving a single dose to the patient. This technology isn't just about understanding a disease anymore. It might just be the key to conquering it, one chip at a time.